because this carboxyl group and the amino group impart significant amount of polarity to the amino acid. Okay. Now, the other thing that I'll just mention briefly, then we can study in the next class, that the conformational feature of this alanine and glycine is also very, very different. Any idea why?
Sir, could you repeat the question once?
The conformation, the structure of these amino acids when they are present in a polypeptide chain is also very different. Okay. I mean, well, you may or you may not answer this question.
Does it depend on like something like the Ramachandran plot?
Oh, you know about Ramachandran plot? Well, maybe it's just you who knows about Ramachandran plot. Okay, we'll look into Ramachandran plot later, but we will definitely look into it. Yes, but the question was not about the Ramachandran plot. Ramachandran plot is just a depiction of the conformational
Yes.
system, right?
So is it due to the non-covalent interactions?
Certainly. It is certainly due to the non-covalent interaction, but don't worry. When we look into the peptides, we will study that. Okay. So something to note is glycine is this alpha carbon is achiral, right? I believe that all of you can appreciate that this is achiral. So this is the only amino acid, only canonical amino acid where the alpha carbon is achiral. Okay, whereas all other amino acids, the alpha carbon is actually chiral. All right. So now if these are chiral, you can imagine that obviously you can have two enantiomers of these amino acids, whereas this glycine, of course, you can't have any stereoisomer, right? Okay. Now so alanine, glycine is the simplest amino acid. It becomes a little more complex by substituting the hydrogen with the methyl group. Further substitution leads to even more complexity. Okay. So for example, so instead of this methyl group, you can have, look at this guy. So this is nothing but this is an isopropyl group which is directly attached to this methyl group, right? This is an isopropyl group. CH3, which is connected to CH, right? Now this guy is called as the leucine. And it does not require enormous amount of imagination to understand that this molecule should be also very hydrophobic. It's even more hydrophobic than alanine. Okay. Likewise, see there is a little bit difference between. So, for example, if you have an isopropyl group here, and imagine this metal instead of connected to it now. If you count the or if you are interested in the nomenclatures, as we discussed, that the first carbon that is connected to the carboxyl group is the alpha carbon. Okay. Accordingly, you have the beta, gamma, and this will be delta 1, delta 2. Okay. So this is the beta and this is the delta. Now you see that here this amino acid can be called as a delta branched amino acid, right? Gamma branched amino acid. Alpha, beta, gamma. Now, if the branching occurs at beta, and there is possibility that branching occurs at beta, look at this molecule. This molecule is called as isoleucine. Okay. In isoleucine, what you have is the methyl group which was present in the gamma position. Instead of the gamma position, is just present at the beta position, right? So these two amino acids, you can say that these are positional isomers, right? These are isomeric amino acids, isn't it? Only the position of the methyl group is altered. But you know what? When we will study a little bit about the structures of proteins, you will see that the beta branch, only by virtue of the presence of the methyl group at the beta position, has completely different properties in terms, completely different conformational properties. Not so much of physicochemical properties, but more of conformational properties than this amino acid, which is leucine. Okay. Likewise, you have another amino acid which is also beta branch, and that amino acid is called as a valine. In valine, what you have is it's very similar to isoleucine, but instead of two CH2 CH3 group here, okay, it's an ethyl group here. Instead of an ethyl group, you have a methyl group. Okay. Now the other thing to note is look at this center, the beta. Is this beta a chiral center, beta carbon?
Yeah.
Yes, sir.
Yeah. So you know what? In nature does all sorts of tricks. Now you have two chiral centers at this center and that center, right? So you have four stereoisomers. And actually in nature you do find all four stereoisomers. And all of these guys have completely different conformational features. Nevertheless, in proteins, if you're talking about proteins that are typically, or a canonical amino acid, then you only have one specific conformation, which is, I think, the S and the R conformation. Okay?
Sir,
Yeah.
sir, are you talking about configuration or conformation? Optical.
Oh, sorry. So, yeah. I mean, hold on. In which case, you mean, did I say conformation of this sentence?
Yes, actually, you were saying that the conformations will be different for isoleucine and leucine because.
Yeah. No. That is conformation. That is not configuration. That is conformation.
So, but then how will the optical activity affect the conformation?
Well, optical activity. Well, see, we are talking about in terms of it when these building blocks are present in a protein. Okay, we are not talking about the individual amino acids per se. If you're talking about the individual amino acids, these two guys probably have the same optical rotation. Probably. Okay. Okay. Can one of you please turn off your mic? Great. Thank you. Okay. So, all right. Now, as we were talking about, then there is another amino acid which is valine, which is beta branch. Okay. Now there are other amino acids which are unbranched sort of. For example, you have methionine, and methionine is a very interesting amino acid because there is a sulfur in your side chain. Okay. And likewise, you can have aromatic amino acids or aromatic residues in the side chain, for example, phenylalanine. And the name clearly suggests that there is a phenyl group, right? Now, there is another amino acid which is very special, just like glycine, which is also very hydrophobic. And that amino acid is called as proline. Okay. Now, proline is a very, very special amino acid, just like glycine. Okay. Now, glycine, see, you can imagine that if you try to, how shall I say? Let's say you have another group here. Okay. Let's not worry about what group. Let's imagine that there is another group which is connected to the NH and another group which is connected to the POH. Okay. Now, if you try to rotate this CN bond, meaning that the hydrogen is, if you're rotating this bond, this will completely go about it, right? Now, depending upon what sort of steric hindrances this hydrogen comes across with the groups that are present in this position or that position, yeah, the conformation of this residue will be determined. However, now look at this guy. Okay. Now, what do you think? Can you rotate this CN bond? Here we were rotating this CN bond, right? As much as we want. Can you rotate this CN bond as much as you want?
No.
No, sir.
What will happen if you try to rotate this CN bond as much as you want?
The ring will break.
Which one will break?
The ring of the proline will break.
Correct. Yes. So if you try to rotate it, what will happen is, you will have enormous torsion at this CC bond or this CC bond, and the ring will break. Well, of course, the ring cannot break because there is no way that we can rotate or induce a rotational force to such an extent that the CC bond breaks. So, of course, that such bonds do not break. But the interesting feature is this is the cyclic nature of this molecule. Now, because of the cyclic nature of this molecule, you can imagine that the torsion, right, the CN torsion, right, the rotation of the CN bond is restricted. Now, since the rotation of the CN bond is restricted, proline has very, 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 very unique conformational features than any other amino acid. Okay, and the other thing that you might note, I will tell you in a minute. Okay. Okay, so these are all the hydrophobic amino acids where the side chains. Okay, remember, hydrophobic, we are considering only the side chains, okay, not the backbone, because the backbone, because the CO and H is very, very polar. Now, you have amino acids which are polar, and out of the polar amino acids, you have amino acids which are negatively charged, amino acids which are positively charged. Now, when are they negatively charged or when are they positively charged? So we are talking about the physiological pH. Any idea what is the physiological pH? The pH in our bloodstream, pH in our body?
7.4.
No, roughly 7.4, right? So what happens at pH 7.4? Okay, so this side chain definitely will exist as a negatively charged residue. What about the backbone? What do you think at pH 7.4 will happen to this backbone NH2 and COH? Will this be charged, uncharged?
The H plus will even, we'll have COO minus instead of COO plus H.
Sorry?
So we will have COO minus, there will be a negative charge on the oxygen.
Right. How about this one?
So there will be a zwitterionic form of the amino acid.
So this will be also positively charged, right?
No, sir.
NH2 will remain as it is.
Sorry?
NH2 will remain as it is.
NH2 will remain as it is. Any
Yeah.
idea what determines whether the NH2 will remain as it is or not?
The acidic strength of NH3 positive. The
Sorry?
protonated.
Sorry?
The acidic strength, pK values
pK of what? The
of
NH2.
the NH2 positive, NH3 positive means protonated form of this amine group.
Yeah, what the pK of the NH2 group will determine whether it will be at a specific pH, whether it will be protonated or not, right?
Yes.
What is the pK? Any idea what is the pK of this NH2 group?
Around 9.
Yeah,
Huh?
9,
So
10.
around 10, 9 to 10, I think.
Yeah,
Yes.
9 to 10. So what pK, what pH are we talking about now? In physiological pH in our body, what is the pH?
So pH is 7.4.
So what is the
7.4.
pH of this?
It will protonate.
So, right? At physiological pH, this will be protonated. This will be carboxylate form, and you also have a carboxylate form. Okay. Anyway, so there are two negatively charged amino acids. One is called as glutamic acid, which is this. You have two methylene groups intervening the alpha carbon and the carboxylate, and you have another amino acid which is called as aspartic acid, which is you have intervening only one methylene group, okay, between the carboxylate and the alpha carbon. Okay. Now, although these groups, they are sort of identical. Physicochemical properties are, you may imagine that they are identical, but they are actually different. They are very, very different when they are put in a protein environment, okay? Only because of the proximity of this carboxylate group, which you can see here, and this group here, okay? Right? So they can have very, very different features. Okay. Now, we also have amino acids which are positively charged at physiological pH, right?
Excuse me, sir. Excuse me,
Yes,
sir.
yes.
So like we have that aspartate,
Yeah.
sir, it has the pH like 9 to 10, and our physiological pH is 7. So like I wasn't able to follow up. Like I don't know. Like after that, I don't understand after that.
You did not understand after that. Which part you did not understand? The pH
Like,
part?
no, we were like we established that the physiological pH is around 7, and the pH of this aspartate is around 9 to 10. After that,
Hold on. Which aspartate is 9 to 10? Which group are we talking about?
so this one,
Which one?
aspartate,
Huh?
aspartate one.
What is aspartate? I mean, you have to name a functionality, right? Aspartate, there is a carbon, there is, CH, there is NH, this is OH. Which one are we talking about?
I don't know, sir.
You don't
Like,
know?
I'm very confused. Like,
You're confused?
yes, sir.
Okay. What is the base?
Base, like NH2
So why is it called a base?
like it has that lone pair and has lone pair, so it can give lone pair, so it can act as a base.
It
So
can
like
donate lone pair, and that's why it can act as a base.
as a Lewis base.
Okay. So yeah, you have to be precise, right?
Yes,
Okay.
sir.
Now you tell me, any idea about the pKa of this NH2 group?
No idea, sir.
Oh, you should read it out then. You should
Okay,
have
sir.
studied. This you should have studied. What about the carboxylate? Okay, forget about this. Do you know acetic acid?
Yes, sir.
Now, what is the pKa of the carboxylate group in acetic acid?
Like pKa. I know the pKa value of that acetic acid part.
What? How much?
0.8 into 10 raised to the power minus 5, the pKa of acetic acid.
10 raised to the power well, a little high, but yeah, close. Okay. But we call it in a not in so, but in a so we call it in the negative log, right? So we don't say 10 to the power minus 5. We're talking about the hydronium ion concentration, isn't it?
Okay, sir.
The pKa is what? Maybe 4-ish or so. 4 to
Yes.
5, 4.2 something.
Yes,
Right?
sir. Yes.
Okay. So now if the pKa of this guy, imagine that this guy is very much like acetic acid. At 4.2, will it be protonated or not? pH 7.4?
Sir, I don't understand this part. Like how to
At pH 7.4, will this group be protonated or not? So that depends upon the relative pKa, right? So if you have two A and B components, right? And if there is an acid-base reaction, one is an acid, the other is a base, isn't it?
yes, sir.
So we are talking about a solution which has a pH of 7.4. So obviously, so forget about solution. Imagine it's just water, okay?
Okay, sir.
And that water has a pH of 7.4, and in that water you have this solute which has a pH of pKa of 4.2.
Okay.
Now, which one is acid? Which one is base?
Sir, that this one 4-1 acid and that 4 is base.
Yeah. So now, if this is an acid, you would have studied about Bronsted and Bronsted acids also, right?
Yes, sir.
So now you have the conjugate base of this guy,
Yes, sir.
isn't it? So that will be
Yes,
what? Carboxylate, isn't it?
Yes, sir. Yes, sir.
Yeah. So please read up. I'm sure that you would have studied about all these things, even HSFE principle maybe. Have you studied about HSFE principle, guys? Hard and soft acid
Yes,
base
sir.
principle? Yes or no? Look it up. Look it up. You should read. Okay.
Okay, sir.
All
Thank
right.
you,
No problem. Yeah. So now we are talking about so there are groups which are also positively charged, the side chains of which are positively charged at physiological pH. Arginine, right? Arginine is a very fancy amino acid. because you have in the side chain a group which is called as the guanidinium group. Okay, because this guanidinium group, it's written like that because this guy is a resonance structure. Okay, even this NH can involve in some inner resonance. Okay, because of the lone pair of electrons here. Okay, now obviously there are multiple NH here, so which is why this is positively charged. Likewise, you also have another group which is positively charged, imidazole. You would probably study, sure, maybe you'll know about imidazole later in your practicals. Okay, so imidazole is an aromatic group, and such imidazoles are also attached to amino acids. Okay, and such amino acid is called as histidine, and which is certainly also basic. Now, one of the other simplest amino acid is called as a lysine, which is just has a NH2 group. Well, at physiological pH, this is NH3+. Now, so you can imagine that these guys can engage in electrostatic interactions. When they're when suitable environment is present in proteins, right? Not only that, arginine, for example, is a very interesting molecule because there are possibilities of multiple hydrogen bonds. So arginine is a guy where these two NHs can engage in hydrogen bonds, and you can imagine not like this. If you imagine it's more like a V form, then both the NH2s, one will be here, the other will be here. They can engage in hydrogen bonds with this and that. Likewise, arginine can also engage in hydrogen bonds with this NH and that NH, right? Now, so that's why arginine is very special, because hydrogen bonds which is involving this residue and that residue, this NH2 and this NH2 is called as a end on hydrogen bond, and hydrogen bonds involving this NH and this NH is called as a side on hydrogen bond. The other very interesting feature of arginine is very effective neutralization of the charges that are present on the phospholipids. Okay. Now, from the lipids, you would recall that we have the glycerophospholipid, right? Because you have the glycerol backbone, and two of the hydroxy groups are actually lipidated with the fatty acids. Okay, and the other hydroxy group, the primary CH2OH, is involved in esterification with the phosphate. Now, here in the phosphate, you have three oxygens which are free, which can now, so you can imagine that the phosphate, something like this, right? So I don't have the phosphate group here, but so imagine this is the phosphorus. So you have one oxygen, another oxygen, and you have the third oxygen, which is double bond O, and the fourth oxygen, which is O, because it's a pentavalent phosphorus, right? So now what it means that the phosphate group, which also has a very similar architecture, phosphorus with an oxygen and another oxygen, and you also have resonance in that form, right? Because there are two canonical forms in which the electron or the negative charge is essentially delocalized between the two oxygens. So since the architecture of this guanidinium group is very, very similar to phosphate, guanidinium groups can very effectively mask the charge that is present on phosphates. And now you probably might remember in some classes back, we were talking about antimicrobial peptides, okay? Where we were saying that, nature has designed or devised many antimicrobial peptides. And it turns out that many of these antimicrobial peptides have multiple arginine groups, not lysine. Although there are some lysine, but arginine is really, really prevalent. And the answer actually lies in here, because the guanidinium group can very effectively mask the phosphate groups. So that is why arginine can very nicely bind to phosphate groups than a lysine. Did you follow what I said?
Sir, I did not understand how the configurations, the structures in 3D of guanidium and phosphate are complementary.
Okay, hold on. Let me see if I can. Uh, is something coming up? Is my oh, I don't have forward.
Yes, sir.
Whiteboard. Is whiteboard visible?
Yes, sir. No.
Oh, very strange. But I can't. Ah, yeah, it's coming up. Yes. Hold on. Yes. Hold on. Let me quickly draw. Okay, so okay, so C and please don't fiddle with this. Okay, N H N H. Let's say NH2 and H. Whoever is writing, please stop writing. Okay, can you see this?
Yes.
Yeah. So now maybe I could draw like this. Okay. So this interaction. Sorry.
So, but this guanidinium group is
Uh,
planar, whereas this phosphate group is sp3 hybridized, that is
How
tetrahedral.
does it matter? But at a specific point, three atoms can always be in a plane, no?
Yes.
Then, meaning that maybe this is this is wedged and this is dotted, okay? But these three can always be in a plane. Yes or no?
Yes, I got it.
Sorry about the movement here. I really hate this guy. Followed?
Yes,
So
sir.
can you explain how this effective in the antimicrobial drug once more?
Yeah. So essentially, what happens is, now you have the phospholipids that are present on the membrane of bacteria, right? It doesn't, a membrane of everything. Okay, even mammalian membrane, eukaryotic membranes. So what I'm saying is the binding is very effective if you have an arginine group as opposed to if you have a lysine. Because if you have a lysine, you will only have well, this will be a carbon. This will be a carbon, and of course, this won't be there. Although this will be NH3 plus. Okay, so you can see that they'll be binding only with one, but the binding with the other one will not be there. And so for every phospholipids, okay, meaning that even if there is a binding, you will need probably another NH3 plus coming and sort of binding into it, right? But it's very difficult because these amino acid chains are not freely hanging. Instead, they are present on a protein or a polypeptide. So meaning that to neutralize one phosphate group or bind to one phosphate group, you will need two molecules, which is again sterically, improbable. Okay. Whereas if you have the arginine group, where you have this bidentate kind of interaction, that bidentate interaction can very well mask the phosphate group. Followed?
Yes, sir. Got it. Thank you.
Okay, now I will share my screen again. All right, where were we? Here.
Sir, could you repeat the names of the hydrogen bonds that you said? Like between the N atoms?
Yeah, one is end-on and one is side-on. Okay?
Which one is end on?
Yeah. So end is, so the terminal. These two.
N-on and side-on. Oh, okay,
N-on
okay.
and side-on, right?
Yeah,
So,
got
for
it.
example, here in blue arrows, it denotes the hydrogen bond donors. Okay.
Yes.
All right. Okay. So likewise, you also have you not only have positively charged and negatively charged amino acids, you also have polar amino acids which are uncharged, right? So,
Sir?
for example, yes,
Sir?
yeah,
Sir, could you please explain what you meant by end-on and side-on?
yeah. So end-on is something where these two NHs, the terminal NHs, which are farthest from the C alpha, engage in a hydrogen bond. Okay. And side-on is one where one of the farthest and one of the closest NH engages in a hydrogen bond. These two. Okay. It's just terminology. That's it. I mean, simple, nothing complicated. Followed?
Yes, sir. Sir, I had just one more question. In this, in this guanidinium, it is the H's that are engaging in, I mean, hydrogen bonding. Like there is no chance that the N nitrogens can engage in hydrogen bonding.
Well, essentially here, this nitrogens can engage. It's not that they cannot engage, but one must appreciate that this nitrogen, so this lone pair is actually delocalized, isn't it? You follow?
Yes, yes,
So
yes,
since the lone pair is delocalized and
yes,
the donor is of course that lone pair, right? You know that's what is engaged in a hydrogen bond as an acceptor, isn't it?
yes.
So that is why the electron deficiency makes this more of an acceptor. I mean, hydrogen bond donor than as an acceptor.
Yes, yes.
Okay. All right. Okay. Now you also have amino acids with polar groups. Like for example, there are amino acids which have amide linkages in the side chain. Asparagine, for example. This is an amide in the side chain. Likewise, you have another residue, glutamine. So, and they are comparable to aspartic acid and glutamic acid. Only thing is that they are amidated and they are not charged. Okay, but they are polar, meaning that water molecule can very nicely solvate, these groups which are actually dotted encircled in red dots. Okay. Yeah, so also imidazole, depending upon the pH, it can also exist in a non-protonated form, right? Because since this is aromatic, so since this guy is aromatic, you can imagine that this lone pair is involved in a, about the Kekule's rule, right? 4n+2 system, or?
Yes, sir.
Yeah. So
Yes,
you can see
sir.
that if this guy is aromatic, this lone pair, and since this is planar, this lone pair on this nitrogen is involved in a in the delocalization, right? Because then it will make a 4n+2 system. In that case, the lone pair of this nitrogen is available for proton abstraction. Okay, so that is why, depending upon the pH, this nitrogen can abstract a proton. But if the pH is suitable, and if it's more basic, then it can exist in a neutral form. And that is why histidine also shows up in the case of, polar neutral amino acid. Likewise, you also have hydroxy groups. Now you would recall that, we were studying about O-linked glycosylation, right? So O-linked glycosylation is present on a serine and a threonine. So you see serine, this is serine, right? You have CH2OH group, and threonine, instead of a CH2OH, you have a CHOH, and one of the hydrogen is replaced by a methyl group. Okay. Now you might appreciate that this amino acid is also one of those amino acids where you have a beta branch. Okay. Like we had isoleucine, we had valine. Okay. We also have threonine, which is beta branch. And so it means that the conformational properties of these amino acids, when they are present on a polypeptide backbone or a protein backbone, is very different. Okay. All right. Now you also have aromatic amino acids. For example, you have phenol group present. You have amino acid which is phenylalanine, and phenylalanine you don't have a hydroxy group, but you have an amino acid which is called as tyrosine, Now, tyrosine is something, where you have a hydroxyl group and you have another amino acid, which is tryptophan. In tryptophan, you have an indole group. Now, something useful for you guys to know that, amino acids, you will see that you can quantify amino acids because of their absorption max. And, there are two amino acids which enormously contribute to their absorption. And those two amino acids are this tryptophan and tyrosine. Okay. All right. Because, it's not very difficult to imagine because of the resonance form. Their lambda max values are much higher than if you have phenylalanine or any other amino acid. Okay. So these are the two amino acids using which you can quantitate or quantify the concentration of a protein. And there are some other features. For example, indole or the tryptophan also has a fluorescent property. And using this fluorescence property of tryptophan, many people use tryptophan as a fluorescent handle, depending upon the usage. Just important for you to know that these two amino acids contribute to the absorption max of the proteins, and also this guy is particularly this guy has fluorescent properties. Okay, now this is all about the amino acids. Now let's come to peptides. How are we doing with time? Anyone?
Sir, we have 15 minutes left.
We have 15 minutes. Good. Yeah. So now, so in nature, what happens is these amino acids don't stay as amino acids, right? They form polypeptides. Now, so what happens is if you have one amino acid and another amino acid, do you think they will just react to form an amide bond? Now, this is an amide bond. All of you know what is an amide bond. It's a CONH functional group. It's called as an amide bond. And when it's between two amino acids, this is called as a peptide bond. Okay. Now, any idea? If you bring two amino acids, will they form a peptide bond? What do you think?
No.
Why?
No.
Because they're ionized already.
Yes, good. Okay, fine. If they are not ionized, if they are in neutral form, will they form a peptide bond?
No, due to
No, sir, an acid-base reaction will take place.
Sorry.
The acid-base reaction will take place.
Good. Yes, exactly. So you will have one acid and one base. They will get into an ionized bond. That's it. So this is not just removal of water. You need catalysts or reagents to do this reaction. And in nature, this is done by ribosomes. Okay. In later classes, you will study about ribosomes. What is the structure, what is the chemistry, in the formation of these amide bonds. Very simple reactions take place by how these amide bonds form. But we'll just, for the sake of simplicity, right now we'll just worry about the formation of an amide bond. Okay. Now it forms an amide bond. Now there are certain features of amide bond. Now if you look into the amide bond, this amide bond, it turns out to be planar. Okay. Now why is this planar? The reason is because you have a canonical form. There is resonance stabilization of this neutral form. Because you have lone pair of nitrogens here, the lone pair can happily delocalize onto the CN bond, and when it delocalizes to retain the tetravalent nature of the carbon, the oxygen, sort of withdraws the electron on itself. So you have a C double bond N and CO minus, okay? And this is the canonical form. So O single bond C and double bond N plus here, okay? Where the negative charge and positive charge is separated. Okay. So now if you have a resonance, meaning that the road or the CN bond. Now since in the current figure, what you're seeing, the CN bond is a sigma bond, right? It's a single bond. Since this is a single bond, it can freely rotate, no problem. Likewise, for example, this C alpha C, it's a single bond. This can also freely rotate. Also, you have the NC alpha, right? Or either this, you can consider this, or you can consider this. Now, since we are considering a peptide unit, so we can consider this C alpha C bond, C O N bond and N C alpha bond. All these three bonds are free to rotate, but because of the resonance stabilization, this bond gets a partial double bond character. Now, if this gets a partial double bond character, you know that you have orbitals. You have the p orbitals, which is above and below the sigma bond, right? And that is why you have a double bond character. And if you have a double bond character, meaning that all these are sort of sp2 hybridized, meaning that this nitrogen, this carbon, this oxygen, this hydrogen, all of these guys are actually in a plane, right? And that is why it is called that amine bonds are planar. All right. Now, then what is it that is free to rotate? Why do polypeptides take some, or proteins take very unique, very remarkable structures? And the reason for that is the rotation about this C alpha C bond and C alpha N bond. Okay, this rotation and this rotation. This rotations, well, they are restricted. We will see how they are restricted, but they are free to rotate because there is no resonance stabilization of these bonds. Okay, and just the combination of rotation of this bond and this bond gives proteins their very unique structures. Okay, all right. Okay, now coming to the next slide. Yeah, so here here this is it's a small polypeptide what is shown. You can consider it's a protein backbone that is shown. And now in this purple color or light pink color, however you may call it. So you have the amide backbone. And in the amide backbone, you can see that you have the amide bond, which is between two amino acids, two different C alphas. You have another amide bond, which is between two different C alphas. Now, if you look into the amide bond, you have the NH, C alpha C, CO, NH, C alpha CO, NH, C alpha CO. It's a repetition of this. Okay. Now, since you have CO and NH in the backbone, right? Water molecules can very nicely interact with the CO. For example, the electrons engage in two hydrogen bonds, right? Because this oxygen can act as a hydrogen bond acceptor for two water molecules. The NH, can act as a hydrogen bond donor for one water molecule, right? So this amide bond is very well solvated. But what is not solvated are these backbones or the side chain. Sorry, not the backbone, but the side chain, right? Here, for example, okay. Now this is the real structure, real three-dimensional structure of a of the peptide backbone, and you can see that here. So this is the C alpha, this is the CO, this is the N.
So your voice is breaking.
Is it? Hello?
No, it's clear.
For me, it's clear.
Okay,
Yes, sir, it's fine.
Yeah, okay. So what you can see that, now, well, I'm sorry, no contact teaching, I cannot show you, or else I would have shown you models. You try to imagine there is a plane, and the plane can cut very nicely through this C alpha, this carbon, this oxygen, this nitrogen, and this C alpha. All these guys are essentially in a plane, but what you do not have in a plane is this C alpha, C beta bond, right? Likewise, this C alpha, C beta, right? This C alpha, C beta is above the plane, and this C alpha, C beta is below the plane. Otherwise, you can have a very nice plane which can nicely cut across all these things. And that is what is planar. So meaning, likewise, so you have a plane which involves this carbon, this oxygen, this nitrogen, this carbon, right? Okay. You have another plane which cuts across this carbon, this oxygen, this nitrogen, this carbon, right? So you have two planes sort of, meeting each other like a V shape. Did you understand what I said? So this will be one plane and this will be another plane. And above and below you have the C alpha, C beta, C alpha, C beta, C alpha, C beta. Is it clear so far?
Yes, sir. Yes.
Okay, good, good. And you
So,
know what?
so,
Yeah,
so
yeah.
can you explain the V part again, the V shape?
Okay, yeah, yeah, yeah. Okay, so just look at my laser pointer. Okay. So imagine I can't draw a plane here, but I can show with a line the plane. Okay. So a plane which is through this C alpha, this COC, this oxygen, this nitrogen, and this, meaning that there will be a plane which you can very nicely fit all these atoms, 1, 2, 3, 4, 5, all of these are in a plane. Is that clear so far?
Yeah. Yes.
Okay. Now let's look at the other amide bond. You have another amide bond here, right? This is a CON. H is not shown for clarity. You have another plane where this carbon, this oxygen, this nitrogen, and this carbon, right? So you can have another plane which will be going like this. I'm just showing the edge of the plane because I can't show a plane. Followed?
Yeah. Yes.
So this plane and this plane will intersect each other in a V-shaped manner. Yes or no?
Mm. Okay. Okay. I got it. I got it.
Yeah, yeah. Now what rotates? What rotates is the relative orientation of one plane with respect to the other. See, I said that this C alpha nitrogen is free to rotate. This is a sigma bond, right? So meaning that the plane that you have here, this can actually rotate, right? This can actually rotate in this manner. For example, if this is like this, it can, come, it can be parallel to your eyesight. It can become perpendicular to your eyesight. It can go 180 degrees, 360 degrees, right? If you rotate this.
Yes, I understand. I
Yeah.
understand.
Likewise, okay, fine, no problem.
Okay.
This is something else I'm saying now. Likewise, so if you keep this plane constant, this plane can rotate with respect to the fixed plane of this. Likewise, if you keep this plane constant, this plane, right, this C alpha and this C can rotate with respect to this plane. Okay? So this is what actually rotates. Okay? And these two rotations are called as, this rotation, like this NC alpha rotation, is called as phi, and this rotation, this C alpha C rotation of the CO rotation, is called as psi. Okay. These two torsion angles are the ones which determine the conformations of proteins.
Sir, how are there two torsion angles?
Yes, these are two torsion angles, right?
So there are two planes, so two planes will form one, and one angle will be there
This
between
is
them.
not an angle, this is a dihedral angle.
Yes, sir.
Yeah, this is the dihedral angle, right? So this is the angle between this plane and this plane. Likewise, so this is another dihedral angle. So these two dihedral angles are free to rotate.
So, but if we are considering only two planes, then how can we have two dihedral angles?
You are considering two planes.
So, are you considering?
Yeah, continue. What did you ask?
So, sir, are you considering three planes, like like two have those two have amide bonds, and one is with the valine, the straight bond which is going down?
No. It's not that. There are actually, well, why you might want to take a cardboard box. Cardboard. It's very difficult for me to explain it here. This is you take two cardboard. The very you cannot take two A4 sheets, but some hard stuff, and try to rotate it. It's a little engaged to understand this. Okay, little engaged, and at the same time, if you can see some videos, that will be very useful. It's very difficult for me to explain from a three-dimensional point of view.
Oh, okay, I got, sir. Means one will be an acute angle and the other one will be an obtuse diagonal.
Well, just rotate, okay? It's not that simple. It's not just about acute and obtuse angle, just rotate. So it's rotation of this plane with respect to that and the rotation of this plane with respect to that. In a simple words.
So how many planes are being considered over here?
Sorry?
How many planes are in consideration right now?
Here we have only 2 planes.
Oh, okay. So 2 planes, okay.
Here only we have 2 planes. Just take 2 cardboards and try to rotate and you will get it. Just do that, okay?
Yes.
Along with some video. Talk to your friends also, they'll help you.
Sir,
Yeah.
it is somewhat like that. If we have a book,
Yeah.
then it is V-shaped when it is open.
Yeah.
Then when we tear apart both the pages,
yeah,
and then rotate the pages with respect to each other.
Correct. That's what. This
So
is
there
exactly
two
what
angles are formed.
That's what exactly. This is exactly what I'm saying.
Okay, I understood. Thank you.
Yeah, and these two are attached in a corner-wise manner, sort of. You follow what I'm saying? Because at this carbon, the corner meets.
Yeah.
Okay, then you have actually 2 angles. Try to do it and you'll understand. Okay. All right. Okay. All right. Yeah. So this is what I was saying. For example, it's a very simple representation of the hydrophobic groups and the hydrophilic groups. So if you have the amide bond, of course, the water molecules can actually engage in well, this is not a amide bond. This is a CONH2. Okay. This is an acetamide, in fact, CH3CONH2. Okay. So here, this CO can engage in a hydrogen bond. If it was an amide, you would have only one NH. This NH can involve in a hydrogen bond. So, whereas if you have a methylene or a methane or a methyl group, whatever, these guys, there will be water molecules surrounding it. Okay, there is no problem in water molecules surrounding it, but the water molecules, they themselves will not form hydrogen bonds. Okay, and that's how the polarity of these molecules will be altered. Okay, yeah. So, yes.
In that previous slide, this there you named the 2 angles or something like that. What was
Yeah. What are those?
What are the names that you spoke in?
Yeah, sure. So this angle which you see between the nitrogen and the C alpha, this is called as phi. Phi, Greek letter phi.
Okay, yeah, phi.
Phi. Okay, and the psi. Yes, the Greek letter psi. Okay.
Yeah. Okay.
All right. Alrighty. Now we'll just talk about another amino acid which is very unusual in some properties than any others. The cysteine. Now, cysteine is a polar amino acid which has a sulfhydryl groups in the side chain, SH. Now, 2 cysteines can engage in the formation of a disulfide bond, okay, through oxidation. And if you have a suitable reducing agent, okay, for example, have you heard of mercaptoethanol or DTP?
Yes, sir. We did it in the last experiment.
Yeah. If you have mercaptoethanol, those are also sulfhydryl group, right? In essence, if you have another sulfhydryl group, so this disulfide group can break, and the other sulfhydryl group that is coming in contact, it can engage in another oxidoredox reaction. Okay. So likewise, so proteins are covalently constrained. So these disulfide bridges provide additional covalent constraints to stabilize structures of protein. Now, something useful for you to know is that typically proteins that are present in the cytosol, okay, in the cytoplasm, they do not have the disulfide bonds. The reason is that in the cytosol, it's a reduced environment, okay, and there are very high concentrations of thiol containing molecules, and that is why there can be spontaneous reduction of this. Okay, so the protein won't be able to maintain its structure. Instead, disulfide bonds are found in proteins that are either present on the membrane or those are secreted out, okay, in the environment. All right. Now, the other thing that is useful for you to know that, in general, the prevalence of cysteine in polypeptides or proteins is typically very low. If I'm not wrong, there is only 2% occurrence of cysteines in all the proteins. Okay, so the presence of cysteines is typically very, very low. Okay, so nature does it in such a way that even if you have cysteines in proximity, so that you don't want to engage in the formation of disulfide bonds, probably that's why the occurrence of cysteines is very low in protein. All right.
Sir, could you explain why you said that they're either produced on the membrane or to be secreted out?
Yeah, because in cytosols, there's very high concentration of free thiols.
Okay.
Okay, so
Yes.
so even if you form disulfide bonds, it will be a spontaneous, redox reaction, and that will not be very much liked by the proteins. Okay, and that is why typically proteins which are in the cytosol, they are not stabilized by disulfide bonds. Okay, so the other thing, I'll just I have a few more slides, then we can stop. So the first structure of protein that was solved in 1958 by John Kendrick is the structure of myoglobin. Now, what you see, this is taken from the textbook, but these are actually historical pictures of clay model of myoglobin, and these sausage-shaped structures, as you will see later, these are structures of these are secondary structures of protein which are called as helices, alpha helices. Okay, right? And when you look into the structure of DNA, you will see that the structure of DNA is so ordered, beautiful structure, helical structure, occasional bends and all, but it's a very ordered structure. But here looks like there was no order. It's just pretty random, one wrapping around the other. So so that actually came as a disappointment to many scientists, because they hoped for a very ordered structure, because the structure of DNA was solved in 1953 or so. Only a few years later, the structure of the first protein was solved. But it's very interesting. Now, from an evolutionary perspective, you will see that DNA structure has to be very simple, because DNA is made up of only 4 building blocks: ATGC, okay, adenine, guanine, cytosine, and urothymine. And DNA, the role of DNA is just to hold the information of the protein. The DNA does not really engage in some sort of a catalysis or signaling processes or so on and so forth. But proteins are the guys which actually engage in n number of processes. So that is why it's very important for proteins to adopt very different structures, right? So it's like for example, if you have a Lego, right, the box itself which comes with that, right, inside you have the Lego building blocks. The box is very simple cardboard box which has the Lego building blocks of different shapes and sizes. The box is very simple, it's very easy to understand the shape of the box. But inside the Lego, right, the box does not itself take part in the functions, but the Lego blocks, some of them are oval shaped, some are hexagonal, some are pentagonal, some are cuboid, right? These are the ones which are involved in recognition with each other, and take part in the formation of signaling networks or whatever in the cell, right? So that is why their structure is very unique. So all the structures of these proteins is very unique, and the uniqueness essentially comes from the different physicochemical properties of the amino acids which build these proteins, right? All these 20 amino acids that we studied, these are the guys which actually fold in different environments in the presence of different proteins to adopt such unique structures and give the proteins their unique functionalities. Okay. All right. So yeah, now is the next class recorded? Can I just continue? We have a few more slides and we can finish it off.
So you can continue.
Okay, thanks. Yeah. The other thing, so this is the structure of a protein. I think it's triose phosphate isomerase or something. Don't worry about the name. It's just a protein. It's a globular protein. Okay, globular protein, which is very nicely folded. It presents in a nice shape, solubility, and all. Now, what we have done is they have sliced through. There is a beautiful transverse section through the protein molecule, and what you see here, okay, is the core of the protein. And the core of the protein, these ribbon-shaped structures in deep purple or whatever maroon color is the backbone, the polypeptide backbone, which is the CONHC alpha, CONHC alpha, what we saw. And in yellow are the amino acids, and you can see that some of the amino acids you probably can recognize. For example, we have a phenylalanine here. Okay, we have a leucine here, we have a tryptophan here, okay, we have a alanine here, we have another leucine here. Okay, all the hydrophobic amino acids are typically present in the core. Okay, it's not that the hydrophilic amino acids are not present in the core. So here you can see that the hydrophilic amino acids are also present in the core, but look at the voids. There are large voids. It's the same protein, okay? Only on left, you are seeing the hydrophobic amino acids that are present on the polypeptide chain. And on the right, what you are seeing are the polar amino acids that are present. And this in encircled region, this is the core of the protein. And this encircled, the encircled is the same, okay? And you can appreciate the number of hydrophobic amino acids that are present in the core is a lot more than the polar amino acids that are present in the core. Instead, the polar amino acids, lots of polar amino acids are actually found on the surface. Okay. Now, this is what happens, when proteins fold. These hydrophobic amino acids, they contribute a lot more to the folding than the polar amino acids. Now, the one thing is very unusual. See, we are saying that, okay, there are less number of polar amino acids, but what about the polypeptide backbone? The polypeptide backbone is polar itself, right? You have CONH in the polypeptide backbone. Now, how do we account for that? That is very polar. How is it that the polar backbone is very well present in the core of the protein? The answer is the following. The polypeptide backbone that is present in the core, they adopt very unique conformations like this, right? So this structure, in B is called as an alpha helix. In alpha helix, what you have is the polypeptide backbone. For example, look at this NH. This NH is engaged in an intramolecular hydrogen bond with this CO. Now, if this engages in an intramolecular hydrogen bond, you don't need water molecule to stabilize, to solvate this NH or solvate this CO, right? So this is exactly what happens in an alpha helix. The alpha helix is one. Okay, so typically proteins, when you name a protein, we go from the N terminus to the C terminus. Okay. Now this is okay. Hold on. Where is our N terminus? Yeah. So this is our N terminus, right? So this is the amide bond. You forget that there is an amide bond. If this is the first amino acid, this is the N terminus, and this is the C terminus. Okay. So we are going from this way to the top. Okay, and you can see that this is an alpha helix where the ith CO, if you consider this as the ith carbonyl, the ith CO is engaged in a hydrogen bond with the I plus 4th NH. Okay, so this is the first amino acid. I means, imagine this is the first, I is any number. Okay, this is the first, this is the second, this is the third, this is the fourth, and this NH belongs to this fifth amino acid, right? So I plus 4 CONH hydrogen bond. Okay, and interesting to note is all the NHs are pointing downwards and all the COs are pointing upwards. Okay, this is constant. Okay, this does not alter. All right. Okay, so this is what is shown. For example, this is the this is the chemical structure. This is the real three-dimensional structure. Of course, you don't see such cartoon kind of things. This cartoon thing is essentially a rendition, but what you see is the polypeptide backbone. And for the sake of simplicity, you can imagine it as a helical way, and you can see that all the CONHs except the ones at the termini, right? They are engaged in the formation of intramolecular hydrogen bond, and that is why even if you have amide backbones which are very polar, water molecules do not have access to it, because they are very well solvated by the formation of the intramolecular hydrogen bonds. Okay, this structure is called as alpha helix. Likewise, you also have structures in polypeptides which are called as a beta sheets, right? So beta sheets are such kind of structures. For example, this is one polypeptide chain going from the N terminus to the C terminus direction. There is another polypeptide chain coming from the N to C, and you form, this is an extended conformation. We will study about extended conformations in the next class.
Excuse me, sir.
Yes.
Excuse me, sir.
Yeah.
Sir, the alpha helix part was in the core, sir, and beta, that sheet is also in the core.
It can be. It can be, yes. But in this protein, in this myoglobin, there are no beta sheets. So this is the protein in this is the triphosphate isomers. I'm not sure about if there are beta sheets, but yeah, I mean, in principle, there can be beta sheets in the core, because here, this is the alpha helix and this is the beta sheet. Again, this is that was the three-dimensional structure. This is the chemical structure. You can see this is N term to C term. This is N term to C term. The polypeptide chains are running in an anti-parallel direction. Okay, where the NH and COs, for example, of this amino acid, the NH and CO is satisfied by the formation of a hydrogen bond with this polypeptide, and of this one, this NH and CO of the flanking amino acid are involved in a hydrogen bond with the next one, so on and so forth. Okay, so this chain runs in this direction, this chain runs in this direction, this chain runs in this direction. So all these arrows are running in opposite direction. Okay, so this is called as an anti-parallel beta sheet. Likewise, this is the three-dimensional structure of an anti-parallel beta sheet. You also have parallel beta sheets where N and C are in the same direction. But then, if you have N and C in the same direction, just because of steric reasons, you cannot arrange them in such a manner. So there is a little stagger, okay? And the stagger creates this kind of geometry where the C alpha, the NH and the CO of the specific C alpha engages in a hydrogen bond from the opposite strand, but of the flanking amino acid, the CO of this and the NH of this. Just look into the structure, you will understand, okay? And this is called as a parallel beta sheet. All right. So,
sir, I had one question over here.
Yes.
Yes, sir. When you told us about the stability of the alpha helix, you showed us hydrogen bonds between the CO and NH group. So, how does that extend to explaining the stability of the hydrophilic side chains as well?
Oh, yeah. Okay. So the hydrophilic side chains, if they are present in the core, typically, they are involved in the formation of either salt bridges or intramolecular hydrogen bonds. Okay.
Okay. Yes.
So that's how they are stabilized, meaning that you won't have a free NH3 group, which is not which is sort of which is not satisfied. The charge is unsatisfied and it's just present in the core. If such things happen, the protein core will be very unstable. It has to be stabilized. Okay.
Yes.
Okay.
Thank you.
Yeah. So so this is about parallel and anti-parallel alpha helix. Okay. Well, I mean, I can tell you that typically it's seen that if you have parallel or anti-parallel beta sheets which are aligned next to each other, there is a typically right-handed twist, and this is what is shown even in coiled coils. Coiled coils meaning if there are multiple helices next to each other, there is also a slight there is a left-handed twist, not a right-handed twist. But in sheets, when they are present next to each other, there is something called as a right-handed twist. And this right-handed twist essentially results in very nice structures. For example, you can Google it up and look into the structure of green fluorescent protein. Okay, you will see that there are all these beta strands, and all these beta strands, only because of the twist that is present, all of these guys can essentially encircle and form a very nice cylinder in between which the fluorophore is present. Okay, all right. So we'll stop here. If you have any questions, you may ask.
Sir, I didn't understand the last slide.
Right-handed twist?
Yes, sir, this slide.
Yeah, I mean, well, we won't go into the reasons of the right-handed twist. I'll only say that. So, here, if you see it, or here.